It's due on Thursday. Okay, cool. I don't know what time, but you should be able to tell what time. Get it done before then. Because if you don't get it done and you send me an email telling me you didn't get it done, I won't answer your email. Because you know it needs to be done. And really, you knew all the answers almost, right? We had covered almost everything before then. So you have that, and that's due on Thursday. Really super easy quiz. It's just freaking homeostasis, man. Just knock that out. Get all the points. They'll get harder as time goes on. Okay, so we're going to finish this set of notes today. Basically, where we were last time is we just finished talking about hormones, but my question to you is, why do some people set aside a separate category and call it neurohormones? How are the terms hormones and neurohormones related, and how are they different?
Well, they're hormones, one.
Go ahead. I'm just going to mess with this for a second. Go ahead.
They're both hormones, but one's emitted by a transmitter or by a neuron.
Yeah, exactly. So my preference, it's not important that it's my preference, but I lump them in with hormones for sure, because once it gets into the bloodstream, does it behave any differently, whether it came from a neuron or from elsewhere? No. So you guys remind me of what you said last time. You said that you understood that the speed of response to a hormone was instantaneous or slower. Slower, right? Because it has to be released into the bloodstream. And then, so it takes a little while to turn on, and then it takes a little while to turn off, right? Does that make sense? And that doesn't matter. It doesn't matter whether it was, for instance, oxytocin that came from the hypothalamus or it was estrogen that came from the ovaries. That makes no difference. So a neurohormone is a hormone, okay? And I want you to understand that, because now we're about to talk about something else that starts with the prefix neuro and has entirely different physiology, really. And that is this little guy right here, which is a neurotransmitter. And a neurotransmitter actually has to meet two criteria to be considered a neurotransmitter. The first criterion is it has to be released by a neuron, yes? And the second criterion is that you can plug that in. I'm not going to go all the way over there.
Okay.
And if I do, I'll give you.
Just catch it.
Yeah. I probably will trip. Has to be released by a neuron, and it has to be released right near its receptor. Do you see? Yes? By a neuron right near its receptor. So with that in mind, without me even telling you, do you think that upon release it has a faster or slower response than a hormone? Much faster. Yes? And how about targeted? So how targeted are hormone responses? What do you do with them? Just dump them into the bloodstream, right? And they go as many places as they find target cells. But is this going to cause a widespread effect, this single release of this single neurotransmitter? No, just right there. But it's not local, actually.
It's not local.
It actually is, even though it's targeted, it tends to be extrinsic, and here's why. I want you to look at this example, and I want you to think that the target cell is the muscle cell of my little toe, right? And the neuron, its cell body, is up in the lumbar region of my spinal cord. You guys with me? So the neuron cell body is up here, the neurotransmitter release is all the way down there. It's still extrinsic long-distance communication. So why is it so much faster if it's extrinsic or long-distance communication? Why is this way faster?
Synapses.
Well, synapses are fast, and also what makes the signal go down the axon? It's an action potential, and they are around 250 miles an hour. So even though it's far away, it can still be fast. Does that make sense? Yeah? Okay. So generally speaking, neurotransmitters are used for extrinsic communication. What is their usual goal? What's the usual goal of most chemical messengers? Not exclusive, not always, but usual. What's the usual goal of physiology?
Homeostasis.
Homeostasis. That's usually what they're trying to accomplish in some way, shape, or form. Either immediate homeostasis or longer-term homeostasis. So let's talk briefly about neurotransmitters. So neurotransmitters, they actually don't get picked up by the bloodstream. If they did, they would be a different kind of chemical, yes? They're only secreted into the interstitial fluid right next to the receptor. And they interact with other neurons primarily, or muscle cells or gland cells, right? Those are the effectors. It's hot as hell in here. Are we going to have to move back?
Yeah.
I'm not moving back today. We're all going to deal with it today because I can't handle the 10 minutes it would take everybody to get settled. So I'll ask her for a different classroom. What the hell? So is there an air conditioner in here? Okay.
I think it's that we're
There's
all
something
just
right here.
really cramped together now.
Yeah, stop breathing. Stop generating so much carbon dioxide. That guy's so noisy. Am I that noisy? Yeah, I am.
Yes.
She can attest.
I am too.
Yeah, you are. Okay, so they allow for what we call synaptic communication, not endocrine communication, which is another type of extrinsic control. And of course, like I said, it's faster, but it's shorter lived, right? So you can have a targeted response, a really quick on, a really quick off. Does that make sense? Okay, so examples of neurotransmitters. You're going to learn, I think I teach 22 neurotransmitters over the course of this semester. It's my favorite. I completely nerd out on neurotransmitters because I like talking about drugs. I like trying to figure out why the hell I'm so crazy and if there's any hope for me. Spoiler alert, no. This is it. This is all I get. So we will talk about those a whole bunch. And I know that you've heard of a bunch of them. Dopamine, serotonin, norepinephrine, acetylcholine, all of those. It's really interesting to me because I'm like trying to get a user's manual after the fact for how to work this thing, right? For how to figure out why you might not be an anxious mess and I am, which might have to do with the time to release of, for instance, serotonin in one of us versus the other, or GABA, which is your chill out neurotransmitter. I don't think I have any. I'm constantly chasing GABA. So that stuff is really interesting. And of course, if you can figure out differential levels that manifest in differential physiological environments, then you can start to figure out how you might manipulate it, which is really interesting. So anyway, I love neurotransmitters. And then the ones that are like ubiquitous, almost every cell can release them, but you probably have heard the least about them. These are called paracrine agents, also sometimes paracrine factors. Paracrine is the important part. And these are chemicals that are released by cells to talk to their neighbors. So if I were a cell, a skin cell, and I encountered a pathogen, I don't really need to tell the whole body at this point. I just need to tell my homies, right? So I release chemicals that talk to my neighbors. Now, the neighbors don't have to be all the same kind of cells, but they are all in the same general area. Does that make sense? So this is sort of your visual for paracrine, right?
Almost
Go ahead.
like intrinsic.
Yes, exactly. Paracrine agents are almost always intrinsic regulation, right? Local regulation. So I want you to look at this picture really quickly and tell me what about that picture keeps the chemical messenger from being a neurotransmitter? Why is it not a neurotransmitter?
It's not really
Exactly. It's not released by a neuron. Other than that, right, it is released right next to its receptor. It is, of course, going to be intrinsic communication, though, because the receptors are right here, right next to me. Do you think you need to release boatloads of that chemical to cause a response? Generally speaking, no. You don't have to release boatloads of that chemical to cause a response. How about speed of response compared to, for instance, hormones? Pretty fast. Is it going to be as robust as hormones? Will it be able to make as many kinds of changes as a hormone? No. Does that make sense? Okay. So the example that always makes the most sense to me with paracrine agents is your response to an injury or an infection, right? So if I get hurt, what I might need to tell you guys is, hey, I'm going down. You guys probably need to go through mitosis soon. Or if I get infected, I need to say, hey, I got this one, bro, but I need you to know that there are bad guys in the room, right? Does that make sense? So localized injury, infection, that kind of stuff, that's paracrine agents. So, and this is generally for intrinsic regulation, as you guys mentioned, and they just diffuse straight to their receptors. Almost every tissue in your body has the capacity to talk to its neighbors, right? Almost every cell does. So don't be at all surprised that almost every cell can release paracrine agents to talk to their neighbors. So let's just go back and do a logic question before we go any further. Now we've got 3 different kinds of chemical messengers. And I will tell you that of the 3, one of them tends to be released in very large quantities. Can anybody guess which one and try to give me your explanation as to why you guessed it? Which one of the 3, hormones, neurotransmitters, paracrine agents, probably you like blast the body by releasing it? Go ahead.
Hormones?
Why?
It has to go further.
Yeah, and it has to go, is it directed going to its target cell, or you just like dump it? And it may have 5 different categories of target cells, but if I release really low quantities into the bloodstream and it has to get to the 5 different categories, right? Then the stats are not with you, right? So you kind of have to overdo it with the hormones, right? And you overdo it. That's also part of the reason that it takes a little while to clear out their effect, because they're in the bloodstream in relatively large quantities. But you don't need to do that with neurotransmitters because it's right near the receptor. And you don't usually need to do that with paracrine agents because it's at your neighbors. Are we okay with that? Okay, so not surprisingly, we actually discovered hormones long before we discovered either of the other 2, because they're so high quantity, so high concentration in the bloodstream. Does that make sense? Okay, so now let's talk about some paracrine stuff. Okay, so tons of different kinds of paracrine chemicals, at least 16 different categories of them. No, you won't memorize them. I just want to give you some understanding of them and then talk about one that you mess with all the time. Okay, so first off, growth factors. That was the example that I gave first. If I get injured and I need to tell my homies, you guys need to go through cell division, does that sound like a local problem? Yeah, I don't need to tell the whole body to go through cell division, right? Just you go through cell division. How about blood clotting? Should blood clotting be global?
No.
No, that would be your last day if blood clotting was global. So if I damage a vessel, I need to release local chemicals that will cause the local response, right? So that's local as well. And then our friends, the histamines, which of course you guys know of in what context? Yeah, allergies and antihistamines. Histamines are released by basophils and mast cells. Do you guys remember what basophils are? Do anybody remember? It's a type of white blood cell. Yeah, it's a type of white blood cell. Do you remember what color they stain? Blue. Exactly. They're not blue. They just stain blue with our differential staining techniques. So, and they're the bananas, and never let monkeys eat bananas, so they're not even very abundant, comparatively speaking. So what histamines do is whenever you have a localized irritation, they release whatever would help to get the area clean, like more blood flow, more snot flow, right? More smooth muscle contraction, sneeze, do whatever the hell you need to do, right? So the reason you probably knew of histamines long before is because you know of antihistamines. And antihistamines work like this, right? The histamine is about to come in and make you have a little snot fit right in the middle of the class. You ain't even got a tissue. What the hell are you supposed to do with that? So this, right? That's what my son does. So just a little bit closer, and it actually causes the response. But the histamine in this picture is in blue. But look, he's been blocked by the antihistamine. And the antihistamine actually blocks the receptor site for that paracrine agent of histamine, and therefore blocks the histamine response. And it's relatively effective, isn't it, at blocking the allergenic response. Now, this is important for you to understand because I know a lot of you guys don't like anything related to chemistry, and you don't want to talk about receptors at all. But let me ask you this. Could you actually have made an antihistamine if no one had discovered the structure of the chemical messenger or the structure of the receptor? No. So pharmacy, intimately related to human physiology, right? You have to discover the inherent functionality before you can actually develop anything that will actually interrupt or augment or whatever the functionality. So this is histamines and antihistamines. So histamines are local paracrine agents as well. And then cytokines. Let's just briefly talk about cytokines. Cytokines are released from a whole bunch of different kinds of cells, especially white blood cells. They're part of the body's immune response against pathogens. They can cause inflammation. They can cause an increased blood flow to the area. They can activate the immune system in the area. Now, that's both good and bad. It's good because inflammation is often the first step of healing, but that's sometimes bad because it's also sometimes painful, right? So you have to decide what level of inflammatory response is acceptable, and we're going to come back to that in just a second. So the other important thing about cytokines is that they are one of the paracrine agents that sometimes will be released in large enough quantities to get picked up by the bloodstream. That's not plan A, and let me be clear. Plan A for paracrine agents just go into the interstitial fluid. But if you release enough of anything, it will get picked up by the bloodstream. So let me give you an example. My 13-year-old this weekend, somehow, in the middle of the hottest part of the damn year, got pneumonia. I don't know how it happened. I don't even know what the hell he was doing. I just thought he was being lazy and wanted to play video games all day. But no, he got pneumonia. So what happened in the initial stages is that he had release of probably a whole bunch of different paracrine agents that caused problems with him breathing, and he's got a lot of phlegm formation and all that stuff. So that was in the initial stages, right? But what happens if he's trying with paracrine agents to battle it off and kind of fails over a few days? Then he's released so many paracrine agents, including cytokines, that he gets a global, not local response of fever. Yes? And sedation. Does that make sense to you guys? Fever and sedation. Go ahead. So a global, not local response of fever and sedation. So does that make it a hormone? No. It means that sometimes if you release enough of it, you will get a bigger response, a more body-wide response. And that's what happened. Does that make sense to you? And you've had that experience before, right? Haven't you had something local that ended up causing global responses? But that's not how it started. How it started was local, right? I started with a local respiratory infection, and then I got a fever and sedation, right? Okay, so cytokines are famous for doing that. Have you guys ever heard of interferon therapy? Have you ever heard of that? It's kind of interesting. Have you? Okay, so I'll describe it just briefly to you. This is one of the therapies that they've actually developed for HIV and also for certain forms of cancer. So one of the things that helps a person who does not succumb to HIV and end up getting AIDS with that is that they release paracrine agents that will battle off both the pathogen and the response to the pathogen, right? But one of the things that happens as you go from HIV to full-blown AIDS is that your white blood cells are taken out. So you stop being able to release these paracrine agents, and your immune system can't really do it for you. So one of the things that they've developed is interferon therapy, in which they figured out how to make interferons in the lab because of, again, people understanding the physiology and saying this is an important part that is actually interrupting this whole viral cycle. And so instead of curing you of HIV, which may have been done a few months ago, but only in a certain set of situations, they will give you a whole bunch of intravenous interferons. What does it do? It interferes with the life cycle of HIV, or it interferes with the life cycle of cancer. And we make it in the lab, which is kind of cool, right?
So kind of like a substituted white blood cell in a sense?
No, it's actually a substitute of what the white blood cell would release.
Oh, okay.
Does that make sense? There's also some interleukins. Okay, but they haven't developed interferon therapy for every type of cancer. Otherwise, you would have heard about it, right? Okay, and then the next category are the ones that I know that you have messed with. These are called eicosanoids. Can you say that, please? Eicosanoids, yeah. So eicosanoids, again, we're still talking about paracrine agents. There's a huge variation in eicosanoids, but they all come from something that you should be able to recognize, which is phospholipids. How many cells of yours have phospholipids in their membranes? Every single one of them, right? Every single one of them. So your cells can take phospholipids, and you do not have to memorize this pathway. Don't freak out about that. They can make an intermediate molecule, and then they can end up making these four. These four are eicosanoids, and we'll talk about what they have to do and how you're constantly messing with them. Well, not constantly, but intermittently messing with them. So they're all made from membrane phospholipids and its little intermediate called arachidonic acid. You don't have to remember that. Don't worry about it. So membrane phospholipids here, and then these four. So let's talk about those four. Okay, first off, leukotrienes. Before I tell you, you tell me what kind of cell you think releases that. White blood cell, exactly. So it's a white blood cell. And not surprisingly, white blood cells release lots of paracrine agents, right? Because they're involved in lots of local stuff, right? So leukotrienes are paracrine chemicals that are produced by white blood cells. They're involved in the inflammatory response and the localized immune response in an injured or infected tissue. So like we said, they're kind of like cytokines, They're not as likely to get released into the bloodstream, but they can, okay? So basically, if you smash into a tissue, whether it's smashing into it with a pathogen or smashing into it with a blunt object, then it's going to release these. And these are both good and bad. Good, inflammation is the first step in healing. It brings more white blood cells and more blood into the area, but it can also be painful, right? And it can also cause, and have you ever gotten hit so hard that it's like hot, right? It can also do that as well. So inflammation and immune response in an injured tissue. So keep that one in mind for just a second. And then we've got another two, and these two right here, the bottom two, that top bottom one's hard for you to see. Prostacyclins and thromboxanes both collectively do something related to the prefix throm. This throm or thrombo. Clotting, exactly. Those two are involved in clotting, which is, of course, a paracrine activity that you're going to need to do. Clotting is really complicated, so don't get confused that I already told you 3 different things that are involved in clotting, because there's like 25. So prostacyclins and thromboxanes, those are involved in clotting. So now we have inflammatory response, right? And we have clotting, right? And then we've got prostaglandins. And prostaglandins are this really big category. There's tons of different kinds of them. And they're paracrine chemicals that can be produced by damn near any tissue. So depending on where you get prostaglandins released, you could, for instance, increase or decrease local blood flow, like they may dilate or constrict local arterioles, right? And that could change blood pressure. They can enhance the inflammatory response, both good and bad, right? They can dilate or constrict your bronchioles in response to a respiratory irritant or pathogen. They can contract or relax smooth muscle. They can shrink your nasal membranes, which will make your nose run. They can do all of that. But then, very importantly, these are the biggies for if they are released in large quantities, they will help their friends, the cytokines, to cause sedation and fever, right? Now, sedation is not necessarily homeostatic. It's just sort of a byproduct of like, hey, your body's really busy. Why don't you sit down? But fever, of course, we know is often trying ultimately to get to homeostasis, right? Right. It's not immediately in the time trying to get to it. So now, let's come back to how you have messed with these, because even if you don't do drugs, you totally do drugs, right? So let's talk about pain relievers, anti-inflammatory drugs, and icosanoids. You've probably heard of the category of drugs called NSAIDs, non-steroidal anti-inflammatory drugs. Okay, can you name any of those? Ibuprofen's a good one. Naproxen sodium. Aspirin fits kind of with them. I mean, it has a really similar mechanism. Acetaminophen is similar, but not exactly the same. So I'm really talking, Aleve is naproxen sodium. So I'm really talking about aspirin, naproxen sodium, and ibuprofen right now. So those have to do with this story that I'm talking about. These are non-steroidal anti-inflammatory drugs, and they block the synthesis of many of the icosanoids, and of course you take them as pain relievers, right? You also take them as fever reducers, right? And then they have these unintended side effects. Do you know any unintended side effects that you can get from taking ibuprofen or aspirin specifically? Yeah, so let's go back to that story and talk about that again. So here's how these work, guys. They are cyclooxygenase inhibitors. So they inhibit that. And because they inhibit that, they inhibit one of the major inflammatories, right? Inflammatory chemicals, and one of the major ones that can cause fever. So you can take out the fever production and the inflammation. But they also reduce these two, which are involved in normal blood clotting, right? So you guys know, probably, right, that if you are on a prescription anti-clotting, they call them blood thinners. They're not actually blood thinners. They're just anticoagulants. If you're on a prescription, should you be taking over-the-counter ibuprofen? No, you should not. And what will happen if you run into something and you are taking both of those simultaneously? You're going to bleed and bruise like crazy, right? I'm not saying you're going to die, but you're going to get a little scratch and you're going to like have to spend 2 hours trying to get the damn thing to stop bleeding, right? So, or you can also use them to your benefit. I have a heart problem, both a valve problem and electrical problem. And one of the things that it does is it allows for the potential for blood to pool in my atria for longer than it should, which increases the likelihood of clotting. Does that make sense? So I am supposed to, and hardly ever do, take 80 milligrams of aspirin every day, even though I don't have a headache. Why am I supposed to take that?
Helps in the blood.
to keep the concentration of prostacyclins and thromboxanes low. But what happens when I run into the coffee table in the middle of the night? I bruise like crazy. I look like somebody beat me, right? So does that make sense to you? Okay, so now notice that we didn't touch the leukotrienes, right? We didn't touch those yet. So if you wanted a really, really even more effective, massively effective anti-inflammatory, you would take out all of them. But that has a caveat. Before we get to that, there are also prescription versions of these cyclooxygenase inhibitors. Have you ever heard of the COX-2 inhibitors like Vioxx and Celebrex? Anybody know why they're prescribed? I mean, there's a bunch of different reasons, but any reasons why they're prescribed? Really massively inflammatory condition like rheumatoid arthritis in the early stage, you can get them prescribed, or an inflammatory condition that's really chronic that is not going to be helped with low doses like these. These are the same kind of things, but they inhibit cyclooxygenase 2, which is really, really effective at reducing inflammation, but they also have the same side effects, right? And that should make sense to you. They do. And they're really effective, and they're going to have some unintended consequences. I don't know if you guys know this, but there was a big lawsuit against Vioxx. There were some deaths related to prescribing it with other medications. I'm not saying they're unsafe. I'm saying that they're unsafe in combinations with other medications. So, and some people still are on Celebrex. Okay, so there is a text box in your textbook if you want to read more about the physiology of those COX-2 inhibitors and the anti-inflammatories. But let's talk about what happens if you have massive, massive problems with inflammation that could be quality of life threatening or life threatening. So let's do two examples. Let's say I have a closed head wound, right? A closed head wound, the inflammation alone could kill me, and it could kill me quickly, right? So I need to do a massive anti-inflammatory, right? Or if I have a child with asthma that's so severe that the inflammatory response in the bronchioles can kill the child, then sometimes they will do a steroid inhaler instead of just a bronchodilator. Okay, so let's talk about those. So these drugs that they use that are massively effective anti-inflammatories are really interesting to me, and it's taken me forever to really appreciate this, but they're called corticosteroids or adrenal steroids. They just call them steroid, anti-inflammatory. Do you probably know the brand name of one of them? Do you know the brand name of one of them? Prednisone, yeah, prednisone. So that's probably how you know that one. Why are they so effective? Well, because they actually inhibit this. They actually inhibit the phospholipase A2, which means you have all of that reduction in inflammation plus that reduction in inflammation. Do you see? So they're super duper effective. They, of course, are going to have side effects from just this story, but now if I tell you the nature of the chemical, you can start to fathom why they have so many side effects. Okay. So prednisone and other kinds of drugs that are steroidal anti-inflammatory drugs, those are actually, and listen to this because it's kind of hard to understand. What we're trying to do is reduce the release of paracrine agents, correct? Yeah? When we're doing anti-inflammation. And the drug that is actually most effective that does that is actually a mimic of a hormone that you have. So I use to block phospholipase A2, I use a mimic or an analog of the hormone cortisol. So just stick with me for just a second. It's going to have receptors a bunch of places, right? Because it's a hormone. So I'm going to get this response, and I'm going to get a whole bunch of unintended consequences. Do you know any of the side effects of using, for instance, prednisone for a decent amount of time? Any idea?
You can have, like, for a long period of time?
Yeah, for a long period of time.
You can get, like, autoimmune.
Yeah, so it
reactions.
depresses your immune system because it's constantly taking out the leukotrienes and taking out the other ones. Right? Go ahead.
Swelling.
Yeah, tons of swelling. And that's because it actually impacts your sodium and potassium concentration, which impacts how much water you have in your body. It's a lipid-soluble chemical. It has impacts on your brain. It will make you moody as hell. Okay? And then once we learn about cortisol, and you don't have to understand this now, but just let me go throw it out here. Because cortisol to me, I don't know if it is to Joe, it's the hardest hormone to truly get a grip on everything that cortisol does. One of the major functions of cortisol is to regulate your blood sugar when you can't eat or won't eat. So have you ever been so sick that you can't eat for like 72 hours? How does your brain stay alive? Cortisol makes it eat you. Eat yourself, right? So what cortisol will do is when times are tough, it's going to say, oh, okay, sure, break down some adipose, but also break down your skeletal muscle and dump it into the bloodstream. Use all of your stores of glucose that were stored in your liver and dump it all into the bloodstream, right? So now that's all well and good if it only lasts for 72 hours. But now I want you to think about a person who is on cortisol, a cortisol analog like prednisone for 6 months, right? They're going to lose muscle mass, yes? They are going to, it really drops your metabolism too. Drop your metabolism so you'll have both weight gain and water gain. Do you know that it increases your likelihood of type 2 diabetes? Does that make sense to you? Yeah? And it'll drop your metabolism and you'll gain weight because of that as well, and you'll have all of the mood side effects. So if you're me, you're thinking, why the hell would you ever prescribe that? Ever? Well, because the option is either excruciating pain or death, right? So it's not prescribed lightly, and any responsible physician will prescribe as short a course as you can get away with, right? A short course, and then you change it. And this, like, they run into like problems with parents in which your parents get really pissed off. Like if you have a kid with really severe asthma and his prednisone inhaler really, really helps him, and then the doctor says you can have that prednisone inhaler for 2 months and then I won't refill the prescription for another 4. Can you imagine what mom and dad say? They're like, F you! But what they're doing is they're trying to make sure that you do not trash the kid's metabolism and immune system, right? So they take them off of it and they'll put them on a bronchodilator, and then when it starts getting severe again, they'll put them back on, which is again why everybody should have to take physiology, right, in high school. You should just have to know this. Okay, so those are the adrenal steroids or corticosteroids, and they're really interesting. They're absolutely necessary, and it has been decades that this has been our most powerful anti-inflammatory, and we haven't come up with a better one that does not mimic a hormone yet. It will be a huge breakthrough when we do, but it hasn't happened. Okay, and then the last category is simply the autocrine agents, and autocrine agents, they don't have a picture of them, but I'm sure you can imagine. Okay, so again, the 4 of us are all the same type of cell, okay? And something happens to me and I release something that communicates with you, right? And you've got a receptor for it, so it's behaving as a paracrine agent. But does it make sense to you that if we are identical cell types, I might also have the same receptor for it? So it could actually act back on me, right? And then it's autocrine. Does that make sense? Okay. So did I release it to talk to myself? Probably not necessarily, but can it do that? Sure. Okay. So for me, I usually lump paracrine and autocrine together, because usually if it can be paracrine, it can often be autocrine. It's not a hard and fast rule, but it is. Okay. So now, are there any situations in which 2 cells can communicate without releasing anything into the extracellular fluid? Yeah, there are, and you learned about them in anatomy. And that is when 2 cells are right next to one another, they can communicate without asking anybody's permission, right? So now I'm going to make you guys all cardiac muscle cells. And you're going to be stuck together, right? And if I stimulate her and tell her to contract, but you have 2 channels that are connected to each other, then you will tell her to contract and her to contract and him. Does that make sense? No. Contract, woo! I usually make people do that, but since you're hot today, I didn't want to make you wiggle. Now, gap junctions are what they call them. I don't know where the hell the physiology book came up with the word connexon. That looks like a Lego word to me. Because we all learned them as gap junctions, didn't we? We don't need a new word. You can call them that if you want, but I'm not going to do it. So that is a gap junction is what it is. Okay, so last but not least, and then we'll get to the next set of notes. How do you know when you run into a chemical messenger what it is, whether it's a hormone or a neurotransmitter or a paracrine or autocrine agent? Don't do what most people do and what I did when I took physiology and memorize dopamine is a neurotransmitter. Cortisol is a hormone. Acetylcholine is a neurotransmitter. And prostaglandins are eicosanoids, because that's not always true. What is true is that any chemical can serve the body in any purpose. So what you have to look at is not what the name is, but what it's doing. So if I am a neuron, I dump dopamine, and you're right next to me and it interacts with your receptor, then it's a what? It's a neurotransmitter, right? But I can also dump dopamine into the bloodstream.
It's a hormone.
Then it's a hormone. Or I can use dopamine to communicate with my non-neuron neighbor, and I'm not a neuron either, and then it's a paracrine agent. And the body reuses chemicals. Once it knows how to make a chemical, it can make it and use it for any purpose. So for instance, you guys know ATP is the energy molecule, right? But guess what? It's actually released in the brain as a neurotransmitter because the body says, I do what I want, right? You just release it, cause a response, reuptake it, and use it as ATP as an energy molecule. So instead of memorizing, you look at the process. Physiology is processes, okay? And yes, most of the time, dopamine is a neurotransmitter, but not all of the time. Good? Okay, all right, so what's this rabbit hole part? That's the little guy looking down the rabbit hole. This is me, that I just never stop reading about anatomy and physiology because I'm a super nerd, and things that you might or might not be interested in. Sometimes it'll be an entire book. Sometimes it'll be a podcast. Sometimes it'll be a video. Sometimes it'll be those kinds of things. Will I generally test off of them? No. Will sometimes I talk about them and appreciate that she is the one person who's nerdy as I am who will talk about me with this crap? Yes. But your time is limited. I can't make you look at every single thing I think is interesting, right? So I'll put those there. But I had to figure out, like, how do I make sure that they could have them if they just wanted them? And my husband's like, nobody wants them. And I'm like, shh. Shh, be quiet, Mike. Other people think I'm fascinating. He's like, which ones? Which people? Which people, sweetheart? Okay. No, we have a good relationship. We're fine. We pick on each other. Joe has met my husband many times. Okay, so now we are switching to chapter 3. I make this as short as I can because I only do it because it's absolutely necessary. Okay, so chapter 3 is cell metabolism. And I know full well that you have had this both in 1610 and to some way, shape, or form you've had it in chemistry. You probably even had it way back in 7th grade. And you probably, most of you, tried really, really hard not to understand it. You're just like, I'm going to write this crap down and then nobody's ever going to talk about it again. And I understand because I did exactly the same thing. I thought eventually people would stop trying to torture me with how the hell to make a protein out of an amino acid or what cellular respiration was all about, but it's never going to happen. So what I'm going to try to do is to take stuff that you've heard before and give you the human physiology spin on it. So don't just listen to, this is the 17th time you've learned enzyme functionality, but how it applies to human physiology. Don't just listen to me talking about cellular respiration, but what it has to do with human physiology. Because I promise you I would not be doing this lecture if I didn't have to, but I do have to. So I'm going to listen to the whys instead of the whats. The whats you've already learned. Okay, so also please note, there is a relatively long before you begin section right here. I'm not lecturing on that stuff because you don't want to hear it and I don't want to do it. Okay, so we will go to now just starting with how I'm talking about metabolism. So when I talk about metabolism, am I talking about how when I was 25 I could eat anything I wanted without getting fat? No, although I really wish I had appreciated that at the moment. It was delightful, and I'm sorry, past Melanie, you were smoking hot, everything about you is perfect, have a sports car. But no, I didn't appreciate it back then, and you won't either. But what I'm talking about is metabolism at the cellular level. Metabolism at the cellular level is the sum of all of the biochemical reactions that are going on inside that cell, including both those that are causing the synthesis of organic molecules and those are causing the breakdown of organic molecules. Does that make sense? Both the making and the using. Both the anabolic reactions and the catabolic reactions. Do you remember anabolism and catabolism?
Mm-hmm.
Anabolic reactions. What does that do?
Builds.
Yeah, it builds things. You generally take small organic molecules and you use energy, right? Because you generally need energy to build something larger. Protein synthesis is a fantastic example of an anabolic reaction. Small amino acids make a big protein. And have you ever heard of the term anabolic steroid? Anabolic steroid, you don't want a catabolic steroid if you're trying to get swole, do you? No, you want an anabolic steroid, right? Okay. So, but don't take anabolic steroids. We'll talk about that later. I mean, unless you need them, and then do, but generally don't. But catabolism is taking an organic molecule and breaking it down to smaller molecules, usually organic, and releasing energy as a byproduct of the breakdown. That's catabolism. Can anybody think of a fantastic example of a catabolic process? I'll tell you, it's one of the three things that's always the answer in physiology. Cellular respiration, exactly. Taking glucose, a large C6H12O6 or some other sugar or whatever, and breaking it down to smaller organic molecules plus ATP. Break it down to carbon dioxide, water, and ATP. Does that sound right? So you need to remember these just because we end up talking about, when we talk about anabolic steroids, you need to know what anabolism means. And guess what? Cortisol is a catabolic steroid. Because what did I tell you it will do when you are not eating? It's going to eat you. It's going to break down your skeletal muscle, right? It's a catabolic steroid. All right, so keep those terms in mind. Now, just a few physiological reactions, not because I'm going to test you over these, but just because when I later say phosphorylation, I want you to have some meaning to it in your head. So I'm not going to make you balance these. There will be no balancing equations in this class. So hydrolysis and why you care about hydrolysis. When water reacts with a molecule breaking a bond. And biochemists would completely say that's not what it is, Melanie. But we ain't in biochemistry. You guys can do that later, okay? We're trying to get the physiology. So what happens is, for instance, you take something with a bond, A bonded to B, you add water, and the hydroxyl goes to one side and the H goes to the other side, right? And you broke it down. Why the hell do you care? Because hydrolysis of proteins is how you break them down to amino acids, right? So that will matter because, of course, physiologically, can you actually absorb a protein? No, you have to break them down to an amino acid before absorption. Does that make sense to you? Yeah? What lab are they doing this week?
3, the biomolecules.
Biomolecules. I did this on purpose, right?
Yep.
Hydrolysis. So Joe says you have to listen.
Yep.
Yeah. Okay. Phosphorylation and dephosphorylation. Okay, we're going to talk about these again today. A really common way to turn an enzyme on or off instead of just making it when you need it, which is slow, right? Does that make sense? So what if I needed an enzyme at a moment's notice, but I don't need it all the time? Well, I make it, and then I have a mechanism of snapping it on and snapping it off, right? So what you do with this one is a very common, but not the only way, of turning enzymes on and off is to slap a phosphate on, right? Let's say that turns it off, and then to yank it back off to turn it off, right? Does that make sense? So phosphorylation and dephosphorylation. Does anybody remember the names of the categories of enzymes that do that? Do you remember what an enzyme is called that phosphorylates another protein? Starts with a K. Exactly, kinase. And an enzyme that yanks the phosphate off? Starts with the phosphatase. So we'll come back to those today. So phosphorylation and dephosphorylation. And then I know how you guys feel about oxidation reduction reactions. So I'm not really going to do them. I'm just going to tell you that they're going to be related to same things today. So this is an oversimplified definition. Generally speaking, you use oxygen. You don't have to, but on this planet, we've got a whole bunch of it. So it's a really good thing to use for oxidation reduction reactions. If I remove electrons from an atom or molecule and slap them onto oxygen, I've oxidized it. If I accept electrons, I've reduced it. And usually the final electron acceptor in cellular respiration is oxygen. So why the hell do you care? Simply because cellular respiration is a good example of a series of oxidation reactions. The oxidation of glucose to water and carbon dioxide. And it only works really well if you have oxygen. Okay, so how am I going to test you over these terms? I'm not really. What I'm going to do is use them later, right? And when I say hydrolysis, you're going to say, okay, I vaguely remember you use water to like break something apart, right? Okay, so that's what I need you guys to understand. All right, so now let's briefly talk about metabolic pathways and then we'll get into enzymes.
Melanie, going back real quick, I'm having a tough time with phosphorylation and dephosphorylation.
We'll talk about it again, so hold on to it, and I'll tell you when we're talking about it again, because we're going to use an example of it today. All right, so you ready? So metabolic pathway, we're going to use this sort of as our starting point today to talk about enzymes, and we'll talk about cellular respiration. And, you know, let me acknowledge, This is my least favorite lecture to do the entire semester. So if it's your least favorite lecture, at least we're on the same page. It's just an absolute necessity to do. So here we go, and then the next time will be more interesting. All right, so a metabolic pathway is simply a series of enzyme-catalyzed reactions to get you the product that you want. So inside the cell, generally speaking, a product is not produced by one chemical reaction, but by a series of chemical reactions. You want to see a metabolic pathway that you don't have to learn so that you can be grateful that things could be worse? Ready? There's one.
Science bitch.
Yeah, oh, that was for my son. He was doing his homework last night, and I was like, I sent that to him. I was being supportive, man. That's a good mom support of a teenager, don't you think? Okay. So each one of these steps, right, is catalyzed by an enzyme. Do you see them? And this is just glycolysis. And FYI, you do not learn glycolysis in this kind of detail in here, but you do in biochemistry. So at one point in my life, I used to be able to draw every single step of this. And do you think I can do it right now? Hail to the no, I can't. All right, so that's a metabolic pathway. But let me just simplify a super, super simple metabolic pathway. Ready? And it's just a fake one. So if what I have is A plus B, it doesn't have to be real chemicals, and it goes to C, which dissociates to D plus E, which combines to form the final product, which is F. Everybody with me? Each one of these reactions will be catalyzed by an enzyme. Enzyme 1, enzyme 2, and enzyme 3. Everybody good? Okay. All right, so this is a metabolic pathway. It's a series of steps, right? And we're going to use this to kind of understand enzymes today because we've got 3 different enzymes, and we're going to focus on enzyme 1 for a minute. All right, so let's talk about enzymes and how you have learned them before and the fact that that didn't help me at all when I was learning enzymes. So here's how most people learn enzymes. They say an enzyme is a protein that lowers the activation energy for a chemical reaction. But I don't know about you, but it always confused the living hell out of me because sometimes they would say, okay, this is an exergonic reaction, meaning it doesn't require energy, it actually releases it. Why the hell do you need to lower the activation energy? Well, they were using the term energy in 2 different contexts right there. So it didn't help me. It didn't help me at all until I got to grad school and one of the professors presented enzymes in a way that actually made sense to me. So if we just look at this reaction right here, we've got A and B, and we want to make C, okay? He explained to me, and it's stuck ever since then, that what an enzyme really does is make it more statistically likely that the reactants will come together, and because it does that, it increases the amount of product that is produced over a given time period. So let me show you what he's talking about. He says that if A substrate has that shape and B substrate has that shape, and they're inside a cell, right? You just wait for them to run into each other and hope C gets formed, right? And then you'll get some level of C at some point, right? But if, instead of doing that, you had an enzyme which had a binding site for A and a binding site for B, yes? Can you see that you have just increased the likelihood that A and B will find one another dramatically? This is enzyme 1.
Mm-hmm.
And therefore, how much C would be formed over the same time period? How much C would be formed over the same time period? Hell of a lot more, right? And if you added more enzyme 1 and there was still enough A and B, you would keep increasing the quantity of C that was formed over and over again. Does that make sense to you guys? So that appealed to me because I was like, I can totally see why that increases the quantity. And it's actually true too, right? It's actually what happens. They have a binding site for the 2 substrates. So let's just make sure that we understand the terminology, and you guys learned this before, but let's make sure that we understand the terminology. For any chemical reaction, right, these are the reactants, and this is the product, right? But when you're dealing with an enzyme-catalyzed chemical reaction, then A and B are not just reactants, they're also substrates. And substrates fit into, what do you call that site? Yeah, they fit into the active site. That makes sense? It's the definition of substrate.
So without the enzyme, they're reactants, then with the enzyme, they're substrates?
If it's an enzyme catalyzed reaction, they are substrates. But regardless, everything on that side of any chemical reaction would be considered a reactant. It's just a more general term. Everybody okay with that? Okay, so now you've known this stuff before, but I want you to think about it in the term of stats and statistical likelihood to make sure that it makes sense to you. So one of the things that I want you guys to understand is that here's the substrate, here's the active site, right? Okay. The product is a different shape than the substrate, right? Otherwise, you didn't do a damn thing to it. It would have still been the substrate. Since the product is a different shape than the substrate, it falls out of the active site, and then you can reuse the enzyme over and over again. Yes? That makes sense? Okay, so does that mean that I made all of this enzyme that I would ever need for the rest of my life when I was an infant? No. Think about it more like durable goods in your home, right? You're eventually going to have to replace your fridge, but not every damn time you use it, right?
Suck.
So eventually, you will need to make more enzymes. So if you had an adult human person who could not make an enzyme in the cellular respiration pathway, whatever you had as an infant would not get you through this, right? Okay, so they can be reused, and they are reused. So everybody okay with that? So they're reused in the process. And so the substrate combines with the enzyme's active site. It's not consumed or changed permanently in the reaction. It can be reused, but not infinitely. Okay, all right, so let's get to some terms that you have learned before and make sure that we can apply them to phys, because we're going to start talking about how this is applying to phys. Okay, so things that can affect, this is what I want you guys to think and try to predict before I tell you. Things that can affect how quickly you accumulate the product C, okay? So start thinking about that. So the first one is a concept that I know that you have learned before, which is called saturation. Okay, but instead of giving me the definition of saturation, I want you to think about what it has to do with this picture. So what is saturation usually? Go ahead.
Oh, I was just thinking.
Oh, you were thinking. I thought you raised your hand. You don't have to do it if you didn't raise your hand. Anybody know what saturation is related to enzymes?
They're all used up in a sense.
They're all used up at the moment, really, right? So what's really happening if this is saturated is, let's say I have 20 iterations of enzyme 1. All of the active sites are full, right? And so look at this graph and make sure it makes sense to you, right? This graph says, and just look at the red line first, okay? This graph says that as I increase substrate concentration, I will have a comparable increase in reaction rate, right? So as I increase A and B, I will have a comparable increase in reaction rate until what occurs? Until all of the enzymes are full, right? And then basically your substrates are waiting in line for their place, right? So it's kind of like a traffic jam. Does that make sense? Okay, so that's called saturation, and that's when all of your enzymes are full. Now, conceptually, what is the only way that you can increase saturation rate? Make more enzymes. That isn't as easy as it sounds, because that's protein synthesis, yes? Right? And sometimes you don't even want to, because maybe you just needed more at the moment. But for instance, if over a long period of time you consistently had an error message that said, I'm not doing enough of this chemical reaction, I'm still not doing enough of this chemical reaction, I'm still not doing enough of this chemical reaction, then might you reset your set point for the acceptable quantity of that enzyme, right? So that's not an instantaneous change in the body. In the lab, it is. When I was doing molecular biology research and you needed more enzyme, I just like squirted it in there, right? But that ain't how it works in your body, right? You have to make some more enzymes. So that's called saturation rate. So the only way that you can increase saturation is to increase the enzyme concentration. So you guys fill this in for me. As enzyme concentration increases, reaction rate tends to increase if you have enough blank. Substrate. Yes, you have to have enough substrate. Because I can keep adding enzyme if I want to, but if I'm out of A or B, I won't get any more C. Right? That makes sense? Okay, so let's go back to a couple things that you may have not talked about before. The concept, and these two are really easy to confuse with each other, so please listen to these. The concept of substrate specificity. Okay, so people have probably told you before, but there is a single substrate that fits an enzyme. And that was only truthy. It wasn't true, right? It's only kind of true. So C right here, and it's not that C, it's a different one. Substrate C is that enzyme's favorite substrate. What do I mean by favorite? I mean that everywhere that this enzyme has a negative amino acid, this substrate has something positive to interact with it. It's exactly the right shape, it's exactly the right size. And it has a really, really good fit, specificity, then how quickly is it going to bind? Really quickly. And then how quickly are you going to get C? Really quickly. Okay, now, C is its favorite. A will work in a pinch if C isn't around, right? But A doesn't bind as well, it's not as specific. So what will that do to the accumulation of C? Decrease the accumulation of C, decrease the reaction rate over time. Does that make sense? Now, on a really, really rough day, maybe D would work, but B, no way, no how. Does that make sense? Okay, now, that means that sometimes there are things that will work in a pinch, right? We have different components in our body. We take in different amino acids. We might have a different version of vitamin D than you needed, but it might work in a pinch, but it won't work as well as the really specific substrate. So, listen to this statement. Substrate specificity implies multiple possible substrates. Do you hear that? Right? Implies that there are, does that make sense?
Can you say it again?
Substrate specificity implies multiple possible substrates. Got it? And I want you to compare and contrast that with the next one that I'm about to teach you, because it's really easy to get these two mixed up. Ready? Okay, so with the next one, they didn't provide a picture, they provided a graph. So let me explain this one to you. This one is, listen, called enzyme substrate affinity. What the heck does affinity mean? How well you like something or attract. I have an affinity for golden retrievers, red lipstick, and dry champagne. All three of those things. They all work to make my life happier. Okay, so, affinity is, let's go two different people. We've got two different people right here. We both obviously can do cellular respiration because we are alive, yes? But we don't have identical enzymes even though we're the same sex and the same species because we're not the same genetics, right? We're not identical twins. We don't have identical enzymes. Now, if she has an enzyme that is actually more efficient at binding to the substrate, given the same substrate, so we're holding the substrate constant here, than I do, then would her chemical reaction be faster than mine? Yes, exactly. So, do you hear the difference? Okay, so enzyme substrate affinity has to do with different what? Enzymes. Hold the substrates constant and realize that not all our enzymes are the same. As a matter of fact, within you, not all of your enzymes are the same because you have two parents, right? And a lot of enzymes, you have two different copies of them, and one of them might work slightly better than the other, right? Does that make sense? Okay, so let's go back again and say that statement that we said before. Substrate specificity implies multiple possible substrates, but enzyme substrate affinity implies multiple possible enzymes. Yes. That makes sense? Okay. Now, do they both happen at the same damn time? Of course they do, but am I going to test you on that part? No, that's me. But of course they do, right? Because she and I have both different enzymes and different substrates. We don't have identical diets. We don't have identical everything, right? That makes sense? All right.
So you said multiple enzymes?
Multiple possible enzymes. Now look, right? So look at the graph. Substrate concentration versus reaction rate. Both of the enzymes will eventually get to saturation, but look at the difference. So this one has a high affinity for the substrate. Look how quickly it'll hit saturation, right? And this one has a low affinity for the substrate, and it takes a little while to get there. Everybody good with that? Okay. All right, so enzyme concentration we already did. Substrate concentration we've sort of been talking about the whole time. But basically, generally speaking, let's go back to substrate concentration for just a second. Generally speaking, as the substrate concentration increases, the reaction rate increases until you hit what? Saturation, right? Until you hit saturation. Okay, and then one last thing that brings us right back to this biochemical pathway. And this one may be counterintuitive, so listen to make sure it makes sense. Okay, so the product concentration can sometimes impact the reaction rate. So listen to this. Okay. So what I have is I have A plus B in the first reaction. via enzyme 1 that gives me C. But then I've got X number, I only put 3 here, but there could be 300. X number of chemical reactions to get to F, which is the final product. Okay? So what I'm going to tell you guys about F is that there is a negative feedback inhibition mechanism that generally interacts with not enzyme number 27, but enzyme 1 or 2. Okay? So let's figure out what's going to happen here. So I'll tell you the basics, and I just want you to tell me what you think it's going to do. So F is actually going to feed back and bind to a binding site on enzyme 1, right there. F is going to bind there, okay? Not the active site. This is called the regulatory site. Regulatory site, okay? And I want you guys to predict, using the principles that we've been talking about in the last set of notes, whether F is going to make A and B fit better or worse.
Worse.
And explain. Try to explain. I'll be nice. Go ahead.
Doesn't it move the enzyme to shift the shape of A?
It shifts the
Active
shape
site.
of the active sites.
Yeah.
Okay. How did you know? I mean, you did know, and you are right. But how did you know that this would be worse and not better?
It's achieved F, and so it doesn't need any more, so that's where the negative feedback comes in. And it's okay, we have what we need, we don't need any to produce anymore.
Right. But this is also based on stats, isn't it? Because I want you guys to think about this over time. So look at this. So if I looked over time, and Jasmine was totally right, and so was Nick, at the concentration of F over time, and I thought about this mechanism right here. This says that whenever I don't have very much F, it's not going to bind to E, enzyme 1, and cut it off. So then I'm going to start making a whole bunch of F, right? F will start to accumulate. And then as F starts to accumulate, it's statistically more likely to inhibit enzyme 1. So F will go down, right? As F goes down, it's less likely to inhibit enzyme 1, so F will go up, down, up, down, up, down, right? Right? Does that make sense? This is a chemical example of negative feedback to accomplish homeostasis, because I would bet you that this is the homeostatic range that we want for F. Does that make sense? But isn't it just based on statistical likelihood? Right? It's just based on shapes and statistics, right? If you have more F, it's more likely to find the binding site. If you have less F, it's less likely to find the binding site. Especially if you realize that we're not talking about one molecule of enzyme 1, we're talking about 1,000 molecules of enzyme 1. So what happens as you generate more F, more of them are off, right? As there is less F, more of them are on. Does that make sense? Okay, questions? Do you see that this is negative feedback? Yeah? And it's controlling the concentration of F over time. So let's fill in that statement right there. Often the product concentration can regulate the enzyme activity. It's negative feedback. Oh, I didn't put a blank there. But you guys predict for me. As product concentration increases, what happens to the reaction rate? It tends to decrease, yeah? Does that make sense? Okay, all right. So now, what we just talked about is an example of this next thing. Okay, so you guys said that you understood that you don't just want to make enzymes when you need them, right? It's inefficient to do so, right? And so what you do quite often is make a bunch of enzymes and have several different mechanisms of turning them on and off. Everybody okay with that? Here's one mechanism. Do you see that that's what I just did? Is turned that off. This is called allosteric regulation, and basically it just temporarily changes the shape of the active site without actually touching it. It's binding at another site, so let me show you a visual on that one. Okay, so what we have is the active site. Let's say this is on. Yeah, that would fit the substrates. And then this is off. And what I can do is I can use some other molecule called a modulator or a regulatory molecule to bind at the regulatory site, right? Not the active site. And by doing that, you can change the shape of the active site. But this is really just temporary, loose, reversible, like ionic and hydrogen bonds, right? Right, so you would have to turn it off over and over and over again with more molecules because it just bind and fall off and bind and fall off, and that's what's going on with this one too, right? But if you had enough of it, it was going to be bound a whole bunch of the time, right? Does that make sense? So this is called allosteric regulation, which is just this loose inner sort of reversible shape interaction. Okay, so this is allosteric regulation. It's a really easy way to turn enzymes on and off. Now, how have you run into this in your life? Well, you have, you just didn't know it. So you know that you need things in your diet, but you have no idea why you need them. Like, you know that you need these trace metals in your diet, these minerals like magnesium and iron and zinc and copper, but you don't know what the hell you're doing with them. You need so little of them that it's obvious that you're not using them to make any organic molecules, right? Not like large quantities to make an organic molecule like protein. So what are they doing? Well, quite often they actually activate an enzyme in a pathway. And if you don't have any of them, then you won't actually get the output that you wanted. But if you have a trace amount of them, you actually will get that. Does that make sense? So that's a mineral. What's the difference between a mineral and a vitamin? They're both needed in trace amounts. Vitamins are organic, carbon-based, needed in trace amounts. Minerals are not. You could get them if you licked a rock as long as it was the right rock, right? Lick limestone, get some calcium, right? Does that make sense? Okay, so these are often called cofactors or coenzymes, and they're used as modulators. And if you've ever been to a health food store, somebody's tried to sell you a cofactor or a coenzyme. Coenzyme Q10, it'll make you not crazy. It won't do a damn thing for you if you already had enough of it, but if you didn't, you might actually see a difference. Does that make sense? Okay, if you just didn't have enough of that trace amount that you needed, then that cofactor or coenzyme might increase your metabolism, might increase your energy level. You don't have any way of knowing, right? Hopefully it won't kill you when you try to experiment with it. Hopefully you're over 18. Okay, and then the same way, the same objective but a different way of accomplishing it, more permanent, is to, instead of just letting concentration bind, fall off, bind, fall off, bind, fall off, we'll do a more permanent change, but it's going to accomplish the same thing. So look at this real quick. Notice that what I'm trying to do is change the active site from that to that. And I'm going to do the same damn thing, but it's going to be harder, slower, and longer lasting. I'm going to change the active site from that to that. But this gets back to your question, Nick. Okay, so this is called covalent regulation, right? And covalent bonds are stronger or weaker than ionic and hydrogen bonds? Stronger. They're harder to make, they're harder to break, right? They tend to last a little longer. So what's going to happen with this one is I have a chemical group. It doesn't have to be this, but this is an example that will work for all semester long. The chemical group is a phosphate, let's say that, right? I'm going to yank that phosphate usually off of ATP because it's always got extras. And I'm going to use an enzyme friend right here to slap it onto this other enzyme, right? And there is how I activated it. You with me? Okay, so if this were a phosphate, this enzyme would be called what? Because it just added a phosphate to another protein. Kinase, yes. Good. Now, you're going to need another friend to do the opposite. It's going to stay on like this until you turn it off, right? And now how will I turn it off? I am going to use a phosphatase. Enzyme B in this example would be a phosphatase to pull it off. But notice it's doing the same thing that we did before. It's just doing it more permanently. It's not permanent because you can still turn it on and off. But again, this one was just based on the statistical likelihood of the modulator finding the regulatory site. The other one was like, you stay on until I tell you to turn off. Right? That makes sense? But they both do the same thing. Questions? No? Okay. All right, so does that answer your question?
Kind of. I'll talk to you. I can
No, don't talk to me after class. I have to go.
No, I'll look more into it.
Okay, and by the way, this right here is not actually a chemical reaction. This is just E for enzyme plus an inorganic phosphate gets phosphorylated, right? And this is a phosphorylated enzyme gets dephosphorylated. So those don't actually represent any like chemical equations or anything, okay? It's just a shorthand of saying it. Okay, so now with that in mind, we are going to do the physiology version of cellular respiration, which you've learned since 7th grade, right? But what I want to do with cellular respiration is instead of trying to teach you the outrageous detail, so if you guys want to do the real version of cellular respiration, and you don't, and I don't want to do it either, the real version of cellular respiration starts here. These are the 10 steps of glycolysis, which is just the first stage of cellular respiration. This little bastard right here is the 8 steps that you do twice of Krebs cycle, right? And this one is electron transport, which almost nobody can draw because you don't know what the hell's going on there. You're just like, it's totally magic. I think there's an oxygen involved. But that is not how we are going to do. We're going to do some science, bitch, but it's going to be the actual understanding of physiology. So you're finished with 1610. Why am I talking about cellular respiration? Because what I want you to understand is, although I didn't have respect for it when I took it, it ends up being sort of something that you come back to in every single body system. So you really have to have a functional physiological understanding of what cellular respiration is going to do. So first off, let me tell you that cellular respiration is basically mostly a series of catabolic reactions in which you take a large molecule like glucose, but you can burn other stuff. It doesn't have to be glucose. And you break it down, but you do it because it releases what? Yeah, it releases ATP energy, okay? And it's arguably the most important biochemical pathway in you, right? Because in the absence of ATP, you can't maintain homeostasis. You spend 60% of your ATP just trying to keep yourself from becoming a flesh-colored pancake. So it is really, really fundamentally important to physiology. But let's just back up and say, it's really cool that you can do cellular respiration, but you are not as cool as a plant. Because a plant can both do the anabolic reaction of producing glucose and the catabolic reaction of making glucose into energy. Because what a freaking plant can do is to stand around and take sunlight with some carbon dioxide and some other crap and make it ultimately into a sugar, yeah? And then it can use that sugar to do cellular respiration inside itself and generate its own ATP to maintain its homeostasis, right? So I just want to acknowledge, as we are destroying every damn thing about the planet, that they are going to be fine without us. We are literally not going to be fine without them because we are not photosynthetic, right? If we became photosynthetic, maybe they'd be SOL, but they're not. So what I want to tell you guys about cellular respiration is I don't need you to be able to do chemical equations like that, but my objective for you guys with cellular respiration is that when we get into a body system, you will be able to relate it back to cellular respiration, right? And that by the time we finish talking about cellular respiration this next time, you will be able to explain the importance of cellular respiration to the human body to a very bright 10-year-old. If you can't do that, you literally do not understand it. If you cannot make your smart child understand why cellular respiration matters, then you have just memorized it and you haven't actually internalized it. So let's just start with the basics of cellular respiration. So you can burn other stuff, but everybody knows glucose, right? And we've already put this, right? You know cellular respiration of glucose. If you want to get the most out of glucose, you need oxygen in order to do it. If you don't have oxygen, you only get 2 ATPs out of a molecule of glucose. If you do have oxygen, you get 36 to 38. Okay, so what you're going to do, and the point of all of this is really that you are going to break this down by catabolic processes and make CO2, which matters. We know it's physiologically important. You don't get rid of it. H2O, which isn't particularly important. This is just called metabolic water. Just mixes with your other water. And then the big one, which is ATP, which I use more than anything to maintain homeostasis. Okay? So by the time we get to your first test, you will be able to explain in 3 sentences or fewer the importance of cellular respiration to human physiology with a specific example, right? So you don't just get to say it keeps you alive. You get to tell me one way in which you would use ATP, for instance, to keep you alive. So let's talk about this. So when I finally started to give a crap about cellular respiration, which was embarrassingly late, right? I literally resisted them trying to teach it to me every damn time. It was when I realized that okay, this is going to make me sound like a blooming idiot, but we all have these idiot moments. Why the hell it was called respiration? Is there any of you guys who did not realize that this is oxygen in and carbon dioxide out, which is literally what respiration is, but this is at the cellular level? So I know it's not just me, and don't even play. I see it out there. You're like, oh, what? But also, show that this is the entire reason that you eat and you breathe. This is the whole reason that you eat and breathe. Well, almost the whole reason. So you eat because that's how you take in these large organic molecules. Yeah? And you breathe because you can't do much with them without oxygen. And you have to breathe because, of course, that's the only way that you can blow off these excessive quantities of carbon dioxide that you are making. So this, the entire reason that you eat and breathe, is to generate ATP to maintain homeostasis. So what you should see is that every single body system, when we get back to it, we will revisit the damn thing. Especially, of course, when we're talking about the respiratory system, about the cardiovascular system when we carry it around, when you're talking about organic molecules and biomolecules in lab this week. So what I want you to do, my objective, is by the time we finish talking about this set of notes, that this actually becomes meaningful to you and not just a memorization. Do you want to know the most common mistake that people make on the first test? Well, I always make you write this. I don't make you balance it. You don't have to put the NADs or any of that crap in there. I just make you write the unbalanced summary equation for aerobic cellular respiration. Guess what everybody leaves off? ATP! You leave off ATP, which literally tells me that you didn't understand the whole point of the damn thing, right? So this is an entire question that you get off if you leave off ATP. Because you just memorized it again, and you already did that 4,000 times, and it didn't mean a damn thing to you the first 3,999 times. Okay, so what we are going to do next time is we are going to go through briefly cellular respiration, and then we'll start talking about the stuff that you're doing in lab. Like, what about starches versus glucose? What about how you burn proteins? What about that kind of stuff? Also, we'll get to talk about the ketogenic diet and all of that kind of stuff when we get into it, okay? I will see you guys on Thursday. Oh, quiz due. Right? You should already have it done. Go stick some answers in there, even if they're not right, and it's done.
Hold more into the fast questions.
Okay, that's fine.
I think I got it. So, pause for